Learning about the cloning vectors in genetics. So here we go. However, the replication will only occur if the DNA contains a sequence which is recognized by the cell as origin of replication. So when we introduce our isolated DNA, what we have selected, it has to have the vector has to have an origin of replication. And since such sequences are infrequent, which are rarely be so, and therefore the DNA to be cloned has to be attached to the carrier or the vector, we can call as, which has an origin of replication. So, what are the ideal vector conditions? So, we have learned about what are the vectors. So, what are the ideal conditions of a vector should have to clone a DNA? So vectors are those the DNA molecules that carry the foreign DNA fragment which I have selected and inserted into it, and a vector must possess some certain minimal quantifications of effective agent of transferase maintenance and amplification of passenger DNA. So the vector should be small and easy to isolate from the bacterium, and they must have one or more origin of replication. So the fragment DNA is easily introduced into themselves and stably maintain themselves within the host cell. And the vector should have one or more unique restriction sites into which the recombinant DNA can be inserted, and they should have a selectable marker, like for instance an ampicillin antibiotic resistance, so we can know the DNA is passed on to the selected vector, which allows the recognition of transmovants. So that's how we know. And vector DNA can be introduced into the cell, and the vector should not be toxic to the host cell. That's And next, what are the types of vectors? The plasmids, they are the extra chromosomals, and they help in the self-replicated and double-stranded closed and circular DNA which are present in any bacterium cell, and the properties are specified by plasmids are antibiotic and heavy metal resistance, nitrogen fixation, and pollutant degradation, and bacteriocin and toxic protections. Some of the plasmid vectors we learn here in the genetic engineering are PBR322 and PB327. And yeast plasmid and TIRI are some of the widely used plant genetic transformation vectors. And there's also a bacteriophage. A bacteriophage can also be act as a vector in the linear DNA molecule single break into two DNA fragments, and the foreign can be introduced into a chimeric phase particle, and which can be capacitated into head of limited, and some of the essential genes may be removed. This technique has been followed in lambda phage vectors and clone large foreign particles. So the plasmid can clone up to 20 to 25 base pairs, kilo base pairs. And the lambda phage, what is the bacteriophage? And cosmids. Cosmid is a plasmid which is certain specific DNA sequences, namely are the cos sites inserted and enabled. Like plasmid, cosmids are perpetuated into bacteria without the lytic development, and the cosmids have high efficiency to produce a complete genomic library. And phasemid, the prepared artificially combining of a features of phages with plasmids. So they are the both, and it's a hybrid of a phages and plasmids. So it's a phasemid, which are P blue script. This PUC19, University of California has developed this, and that's how we can say. And the plant and animal viruses, the number of plants and animal viruses has been also used as vectors, both for introducing foreign genes into the cells for gene amplification. So we have learned about tobacco mosaic viruses, right? So how are they separated from the group of viruses? humanoid adenoviruses and retinoviruses are potential vectors for the gene transfer into the animal cells. So that's how we can also develop the COVID vaccine. They use the adenoviruses of the animals and etc. And artificial chromosomes are each artificial chromosome or bacterial artificial chromosomes can also be act as vectors for cloning several hundred kilo base pairs, which maybe represent the whole chromosome. It can clone in yeast or bacteria by ligating them into the vector sequences. And transposons. We learned about transposons, which carry the genes from one place to another place, which change the genes. So these are the transposons, which also act as a vectors. So thank you guys.